discussing the little background about what we are going to do and what is the importance of our project. And little if we have to study about a infectious organism, it is important to study little pathophysiology. What is its physical structure and what are the different components present in that? All that we have studied. So since being this is my research area of interest, I have done some basic research to identify some pathogenic markers. Pathogenic markers in the sense like based on certain proteins, we can identify that parasite. It can be any organism, whether it is bacteria, fungi, algae, whatever. We can identify a particular organism based on the surface proteins. Directly we can come to the concept based on the surface proteins or based on certain protein composition of the organism we can identify. So that analysis I have already done, and the paper also I shared in the group in the Google Classroom, that regarding identification of a the protein I shared. Right. You are on my screen. All of you are on my screen. Yeah, I'm just struggling a bit. Yes. So, yes. So last class, hope you are on the PPT. Yes. So last class we discussed about a little pathophysiology, how this particular organism looks. So as we have studied, this is a protozoan parasite, almost like same phylum of your malarial parasite, filarial parasites, which cause very deadly infectious diseases. This is also such a kind of parasite that causes sexually transmitted diseases in cattle, leads to what are the different symptoms I mentioned? When this parasite infests the cattle, infests the cow or the bull, it causes damage to the uterine damage to the genital tract, leading to early abortions in cows and very low fertility rate in cows, and so many different kinds of vaginal secretions. So these are all the symptoms. Because of these symptoms, cow will not be able to conceive, and it will not be able to reproduce. Likewise, it will not be able to give a good amount of milk, and same way bulls will not be able to grow well, and they'll not be able to produce so much amount of meat. Altogether, why we have to worry about this cattle is because we depend on cattle for several dairy and other dairy related products, right? So if there is no production, we will be in so much crisis. So to address that issue of low productivity in cattle, this was area to identify presence of several infectious diseases in cattle, and several studies were proposed by governments that some kits need to be developed to screen for these diseases in these cattle, so that we can eliminate these infectious cattle from the herds, and we can prevent the other herds from getting infected. So that is the actual strategy. So in order to identify, or in order to design suitable drugs, so we need to understand little suitable diagnostic kits or suitable drugs for treatment. We need to identify the structural features of the bacteria in little more detail. So I'll share one more paper. You may not understand much, but based on that paper and based on the paper that I presented, so this is my paper. I published this paper wherein I have identified several studies related to this organism, Trichomonas fetus, mostly because I am interested in looking at some proteins, very important proteins, mostly the surface proteins, which are like almost exterior cell membrane proteins, because they tend to be more antigenic and they tend to be more interacting with the surrounding. As well as I'd also try to pick up some proteins that are secreted by this parasite into the vaginal or genital tract and causing the damage in the genital tract. So it's because whenever we have to look for any pathogenic marker, these are called as markers. Based on these proteins, we can identify that particular organism, and they are they should be very specific to that organism. So that is how we have in our paper we have identified and shortlisted few proteins which were found to be present in large quantities. And one more criteria that should be taken, what kind of, because you will have millions of proteins, so you should choose such proteins which will be in higher concentrations present in that organism. Preferably they are present on the outer membrane region. Maybe they may be secretory proteins. So if they are in large number, definitely they'll come in contact with their surrounding. They'll exhibit antigenicity to a large extent. That is one criteria. Second thing is, if they are in the outer membrane, definitely they will have the access. And if certain organisms secrete out certain toxic proteins into the environment so that they make place for themselves, such kind of proteins also are more suitable when you have to look for developing a identification of a diagnostic marker or a therapeutic means such places you can target that organism for killing also, like for treating, as well as the same way you can use such proteins for diagnosis also. So based on such criteria, we have identified and listed several proteins. You can see here almost 10 proteins are there. So, and this is actually the final chart. This part you may not understand right now, but based on the analysis, we have listed out the based on several others research findings, because they have, what research finding was actually, they have analyzed complete protein composition of this organism. Because when you have to study about organism, you should as a previously it was like they used to study one protein and they used to come up with outcome. But now with the advent of bioinformatics and genomics proteomics technology, at the same time we are able to analyze all the proteins, same time we are able to analyze all the genes, all the transcripts, etc. So with the availability of that high-end technologies, now several scientific papers were published wherein they have analyzed complete protein sequences, complete mRNA sequences that are getting expressed. And in from those papers, I picked up certain proteins, certain mRNA that were produced in large quantities. Because if an organism is producing some protein in large quantities, means definitely it will have some survival advantage to that organism. Then only it will produce large amount of protein. Why we have large amount of hemoglobin? Because it's very important protein. It has to carry oxygen and carbon dioxide in the body. Likewise, this particular organism was found to produce all these proteins in relatively larger quantities. So that larger, we have taken a scaling wherein a protein that is produced 1.5 times higher than the normal level. So in those so when it is required, these proteins are produced in more than 1.5 to 1.8 times higher the concentration. Such kind of proteins only need to be taken into consideration, because they may give us a clue to identify a suitable marker, suitable protein, suitable drug target. They can also be used for developing vaccines. So identification of suitable proteins, pathogenic proteins and target proteins are used for developing diagnostic kit, or you can target them with drugs so that you can kill the pathogen. You can also develop vaccines using such targets. So identification of suitable proteins has three advantages, right? So, and based on the data that were produced only, I could get the accession numbers of these proteins. So now the first part I have already demonstrated for you is, you need to find out how big is that protein. How big are those proteins? So how will you find out? So proteins are made of amino acids. They are just polymers of amino acids. They will have N-terminal and a C-terminal. So when I analyzed, so based on the availability of this accession number. This accession number is quite unique, and it is quite unique to each protein that is deposited in the database, in the biological database, that is NCBI. Like how your roll number is unique to you, this accession number that is deposited in the NCBI is unique to each protein sequence deposited by each scientist. So there will not be any overlap between the accession numbers. So using these accession numbers, you can go to NCBI. And so what is NCBI? I told you NCBI is National Center for Biotechnological Information. This is like repository, because previously that very less technological advancement was there. People used to do very less genomics, proteomics, small proteins only they used to analyze. Limited number of genes only they used to analyze. But now, because of availability of next generation sequencing technology, each day so much of sequencing is done. So if they do the sequencing, where they will save the data. So likewise, throughout the world, the globe has come together, and different organizations have come together and collaborated, and all the biological information is stored in at National Laboratory of Medicine and National Center for Biotechnological Information. So here, if you go to the tab, you can get so much of data related to genomes, and so many nucleotides, proteins. You can get data related to your literature and so many things. So this part we will discuss later. And at this point of time, we are interested in getting protein sequence. You need to know the size of your protein, and you need to get the amino acid sequence of your protein, right? So so I will also share this document today, wherein I will share this Word document wherein you will be able to copy the accession number of a particular protein, and you should be able to. So what is the first step? You have to go to NCBI and you have to select protein, because you have to analyze protein. You need protein related information, whether it is sequence or the number, whatever. So you paste the accession number when you select the protein. Just give a search option. So it will take a few seconds almost, and you will be able to identify. So this is the home page of a protein database called as GenPept, wherein you can see this. The name of the protein is cysteine protease 8. The organism is Trichomonas fetus. It is also called as Trichomonas. It is also called as Trichomonas. Both are same. So if you just scroll down the page, you can see very clearly that it has 320 amino acids. And you don't go right now down. So here itself you have a tab called as FASTA. So when you click on this FASTA, you will be able to, you will be directed to your next page, wherein you will get the amino acid sequence. So any FASTA sequence has a is indicated with the help of a greater than symbol. You will have the accession number, name of the protein, and the name of the organism from which it is isolated. And here you can see the amino acid sequence. So you have to copy this and paste it in a Word file. So you have to keep pasting in a Word file. So likewise, this document I will share today, and all of you have to open a Word document, and all of you have to retrieve. What is retrieval? Getting. All of you have to retrieve. So what I'm doing now, I have taken the second, the accession number of the second protein from the list, right? So when I submit, you will get the amino acid sequence of the second protein. Don't worry about if you get little different names. Please do check whether you are getting the accession number your accession number is same with what that is being displayed, because the list I have given is these, because these why so much work is not done on this parasite is that this is like neglected parasite for many years, and recently in the sense almost like 2016, first sequencing was done. Nowadays we are comprehensive understanding is possible only based on the sequencing. So since the sequencing is done very recently, sequencing is done and scientists have deposited, but it is not much analyzed. So certain proteins, they do not have the they do not have much information. So in my research work, I have analyzed this and I have compared it with some other already known proteins, and I have named that protein. Like I gave it like protein. Means something like that protein of different organism. So that is how I named. So don't worry if you get a little different name, but you make sure your accession number matches. Now, so you note down what is amino acid sequence size. Again, you go to FASTA along with the arrow mark. From the arrow mark to down. So you copy and you paste it. You copy and paste it in your new Word file that you will be using. So this, all of you have to retrieve all 10 sequences, right? So now you would like to know little physicochemical properties, what this protein might be, and little more details you may like to understand. You need to understand, because finally you have to choose which protein might be most suitable at this point of time for a diagnostic, for a therapeutic, for a drug target. You should be able to identify. So that is possible only after uh, certain analysis. Little analysis you should do with, because just by looking at the sequence, you all look at the sequence, I'll show you once and you tell me, do you understand anything out of it? Do you understand anything out of this all amino acid sequence? Anybody? Divya?
No.
What can you make out from this? Can you make out anything? You can only make out that they are amino acids. G means glycine, E is glutamine, S is serine. Only this much you will be able to make out. But you will not be able to make out anything. What kind of structure it may have, what kind of function it may have, where it might be present in the cell, whether it is in the cytoplasm, nucleus, where it might be present, and whether it is acidic protein, whether it is basic protein, and so many things you need to know. So by just having a sequence in hand, you will not be able to identify, you will not be able to predict any properties regarding your protein. Because I told you from these proteins, all 10 proteins, you should choose one for the beginning of an experiment. So after thorough analysis only, you will be able to identify. So the first analysis that we will do is to understand the physicochemical properties of the protein, right? So for the sake first protein we have first protein sequence we have a cysteine protease 8. So physicochemical properties of the protein, if you have to do in the laboratory and try to understand what are the physical, you can do in the laboratory also, but it will take lot of time. So you have very good online free resources wherein you can just submit your you can just submit your sequence and you can get the physicochemical properties of the protein. So what is the site is expasi prot param. Expasi prot param is the site wherein you just have to submit your sequence. So can anybody read? So expasi protparam is a tool which allows you to computation of various physical chemical parameters for a given protein sequence, whatever. So it calculates its molecular weight. It calculates its theoretical pi. It calculates it analyzes the amino acid composition of that particular protein. It gives some atomic composition. It gives some extinction coefficient. It gives very important parameter is its stability. It gives some estimate of its stability, whether this protein is stable if it is produced outside. So likewise, several very good parameters it will give, and you will be able to figure out very easily. So if you have to do this experiment in the lab, it will take 6 months. So within 30 seconds, you will be able to get all the parameters by using bioinformatics tool. So what you have done, you have to go to Google. Just you go to Google and type expasi prot param. So you will get this home page. You just have to paste your sequence. So most of the tools will ask you to paste your sequence along with the greater than symbol fast, that is called as FASTA format. Sometimes if they mention, you should see very clearly. Sometimes they may not ask. So let's check. So after you paste your sequence, you just have to do the compute parameters. So it will take few seconds. So looks like here this tool, though they did not mention, we have given FASTA sequence, right? So this is not accepting FASTA format. So it may accept from your first amino acid, that is methionine, right? So let's give, yeah. So your Swiss expasi prot param, you need to paste the sequence that is from starting amino acid to the ending amino acid. It is not taking FASTA. Certain tools will directly, they will ask FASTA sequence. You will have to paste the entire FASTA with the greater than symbol. So now let's look at what are the different parameters you are getting, what are the different physicochemical properties. So the number of amino acids that are present in your protein is 328 amino acids, and you see within seconds you are able to get the molecular weight of your protein very accurate, because knowing molecular weight of your protein is very important, because in the experiments you will be separating your protein, you will be synthesizing your protein. So when you run it in the gels, you should be able to identify whether your protein is produced and where it is matching. So knowing the molecular weight of your protein is very, very important. And you see theoretical pi. What is pi? Pi is the isoelectrical pH at which the protein will have equal amount of positive and negative charges. So this particular component is very useful when you do the two-dimensional gel electrophoresis. You don't need to worry at this point of time. So, but very important parameters. So, among your all 320 amino acids, you can see here alanine has almost like 10%. Composition of alanine is 10%. Likewise, you see all different amino acids and their percentages. And next to this, the most important parameter, then rest all are not so important. But the last parameter from this is the estimated half-life. So what is half-life? Stability. For example, half-life of our RBCs is 120 days. So within RBCs, they stay for 120 days, and later on the protein gets degraded by ubiquitination. So the other day I taught the second years that post-translational modification. So ubiquitation, any protein that is getting ubiquitated means it is going to get degraded into amino acids. So after its half-life, the protein gets degraded. So, why you have to know about half-life of a particular protein? Because you're planning to develop some diagnostic or therapeutic, right? So, you have to synthesize this protein in the lab. When you synthesize it in the lab, or when it is produced in a particular host, how long this protein is going to be stable under normal temperature? You knowing that is very important. So, if you check here, estimated half-life of this protein is 30 hours. When you synthesize this protein in the mammalian cells, it is more than 20 hours when you synthesize this protein in the yeast, and it is more than 20 hours if you synthesize in the E. coli. So relatively, it is a stable protein. Even if you synthesize in the E. coli, you can recover that protein within 20 hours, and you can store it in the by using several storage procedures. So it's a for certain proteins here you will get very directly, you will get that this is a very unstable protein. So such proteins are very difficult to handle in the laboratory. So such proteins really you should not means at that in the beginning you should not handle such proteins. So relatively here you should check some proteins that are stable to synthesize in different, because will not be synthesizing from Trichomonas foetus because they are very difficult to culture. You will not get much protein. We will be synthesizing in E. coli or yeast because they are easy to grow and we can get lot of amount of protein, how much ever we require. So this analysis indicates that they are very stable, and they can be synthesized in the mammalian cells, yeast or E. coli. So this is these are very important findings, and few things you need to note down. So this analysis you will have to perform for each protein. So all the 10 proteins. So what are the different steps? You have to type expasi prot param. Otherwise, you can just type prot param tool. So prot param tool, go to prot param tool and paste your sequence. So since we have already tried pasting entire sequence, this did not take, right? So type, you paste from methionine, the first amino acid to the last amino acid, go for computing parameters. So soon you will get. So what all you have to note down? You have to note down how many number of amino acids, what is a molecular weight. You have to note down for the theoretical PI. And from this, the very important information that you require is the stability. Stability in different organisms. When if you synthesize in mammalian cells, how many hours it is stable? If it is in yeast, if it is in E. coli, how long this protein is stable? So these are very important parameters that you need to learn. And second important analysis. Next, now you understood the molecular weight, acidic, all that you understood. And second interesting thing that you may need to understand very in the beginning is where this protein might be located within the parasite, because these are parasitic proteins, right? So where this protein might be located within the parasite, that is a very interesting concept. So how will you analyze? Again, there is a tool, very free tool available. Within seconds you will get the accurate results, almost accurate results. So the name of the tool is Wolf P Sort. The name of the tool is Wolf P Sort. Wolf P Sort is a freely available tool that tells protein subcellular prediction. What are subcellular? A cell will have a nucleus, cytoplasm, it has some cell membrane and several other organelles, right? So this tool, very free tool. So this tells you, so here again, you have to paste it. It will give the information whether you have to paste it in a FASTA, enter multi FASTA format only it is telling. So you can enter your complete sequence with the accession number, all that, and you have to submit. So here one selection, only one parameter you should check here is this particular protein sequence that you are going to analyze belongs to either animal, either plant or fungi. So now you tell me this particular protein that you are going to analyze in this tool belongs to animal, plant or fungi. Which one we have to choose?
Animal.
Animal, because it is a parasite, right? So only this part you have to choose, right? So then you have to go to submit. You have to give submit. So this will take a few moments. This will take few moments, like few seconds. Yes. Still it is processing. Yes. So here you can see the output given in a very small. I will I will enlarge the screen. Not able to enlarge. Yeah. So can you see the screen? So this is how you will get the output. So this is the accession number that you have submitted, right? So you will get some some short forms, and you will get some values. These values are nothing a comparable values, but within these numbers, can you compare 1 and 20? Which one is bigger? 20 is bigger, right, when compared to 1 and 20. So these are some statistical values related to a particular analysis. You cannot compare this value with some other analysis. So within this analysis, here you can see different subcellular organizations. I have told you, right? You have nucleus, cytoplasm, cytoplasmic peroxisomal, you have plasma membrane, you have Golgi, and you have extracellular. So for this particular protein, you tell me which of the particular location has highest numeric value? Anybody? Your
Extracellular.
extracellular. See, extracellular has the highest numeric value. Highest numeric value indicates the most probable location. Most probable location of that particular organism, particular protein in that organism. So, what is the protein we are browsing now? What is the name of the protein? The first protein. This is the accession number we are using, right? So, what is the name of the first protein? Cysteine protease A.
1648.
So, what our tool has predicted this protein to be? This our tool has predicted this protein to be present most of the concentration outside, means it is secretory protein. And if you anyway, a protein has to be produced within the cell, right? So that is why you see a small portion of it present within the cell component, but most of it is being excreted out of the parasite. So this is how all these predictions will help you to judge, to give you better understanding of that what that protein might be doing within the parasite. So same way, let's also do for another protein. Let's do for another protein. So all this analysis is easy for you only when you when you copy paste all your sequences. So tomorrow what you will be doing is you have to go to computer lab, ask Radha ma'am, and mm, you will have to practice. So now I have pasted the amino acid sequence of the second protein, and why it is not taking, it didn't get highlighted. Still it is not allowing. Sometimes very bigger proteins also it will not take. Now let me give some other small sequence. So you can, when you can work out with all this, only when you have all the protein sequences in hand. Without having the sequences, you will not be able to work out, right? So let me open your protein sequences. Yeah. So here are your protein sequences that I have in my hand. But you have to first download. All of you should have all 10. So here I have taken the FASTA sequence of your a protein from the list, and we'll have to do the. So hope you're following me. I'm not too fast. I'm going little slow only. Somehow this is not taking. And we have to choose. We have to choose animal. I have taken some other protein, and then you have to submit. So it will take few moments. So tomorrow you will have to practice downloading all 10 sequences. You have to do XPASI, and you have to do Wolf-P sort analysis. Yes. So now our, what was the second protein? Let me just check what did I copy. I copied some protein. Just a second, I'll go back. The name of the protein is a subtilisin-like protein from your list. So subtilisin-like protein, let's see. So this subtilisin-like protein is also has highest location in the plasma membrane. Highest amount is in the plasma membrane, little concentration is in the extracellular. So what you can see, this is mostly present in the membrane. It is a membrane protein. So that you can make out. So likewise, you will have to analyze you will have to analyze and try to understand where the what might be the location, subcellular location of the target proteins that I am giving you. So that part you should understand. So after you understand, and as I mentioned in the beginning also, why I have listed only those 10, though there were almost like 1 lakh amount of proteins in that particular organism, is that I found them to be expressed in large quantities, and I predicted them to be present somewhere in the membrane or maybe secreting outside. So now you found two examples. Cysteine protein was extracellular, and this particular protein is present in the plasma membrane, right? So to confirm the location of the protein within the organism, you will have to do one more analysis, is called as TMMHM. So this is also a freely available tool. You will have to just paste your FASTA sequence. TMHMM. You just type in Google, you will get it. And you don't need to search anything, you just submit. You don't need to use any parameters, you just submit. So it will give you results within seconds, and it is very easy to analyze the sequence also. So as I mentioned, membrane proteins can be present, uh, they'll be present in the membrane. So some part will span through the membrane, some portion of the protein will be exterior, certain protein will be interior. So that you can, if you can see here the analysis. So outside indicates outside the membrane. So large amount of this protein, 819 amino acids, is outside. Almost like 22 amino acids will be within the membrane, the double layer fluid mosaic model, right? And small amount of portion is present within the cytoplasmic side. Presence of this violet bar, one violet bar you are able to see, right? It indicates that this protein is traveling through the membrane once. This is called as membrane spanning domain. This is the extracellular domain, and this is the intracellular domain. So what is that you can infer? From this list, we should choose certain proteins that have large amount of extracellular, little amount of intracellular. So that if you choose a large proteins that are present in the extracellular, they tend to interact with your surrounding, they can be antigenic, so they are most suitable proteins. So you also have to practice this TMMHM using, all you have to first download sequences, and you will have to analyze. You all practice tomorrow, and I will tell you how you have to tabulate. So don't worry about tabulation, copy pasting. Your only thing is you have to copy all the FASTA sequences tomorrow, and you practice XPASI, Wolf PSOT, and TMMH. So I'll stop here, and we'll see tomorrow. Thank you.